Hello guys! I hope you've had a great week and that you are ready for another exclusive video. Today we will talk about selective breeding in a bit more detail. We will talk about GMOs, or genetically modified organisms, and lastly we will talk a bit about the genome. We discussed in a previous video how humans have been selectively breeding and modifying domestic and farm animals to adjust to our needs. Our farm animals have been bred to be more tame, to provide higher yields of the product they are bred for, etc. Domestic animals, on the other hand, have been bred for other purposes, to become more aggressive, protective, cute, fluffy, energetic, etc. But as usual, there's a catch, so to say. These animals have been bred so intensively that nowadays, some breeds are almost guaranteed to have certain congenital diseases and other health issues. So for example, you might want a dog that looks very cute and that has a flat nose. To get one, you would breed two dogs that have that characteristic. They both have flat noses. This way, it is almost guaranteed that the babies will also have flat noses. The problem is that two dogs which present similar characteristics will probably present similar genetics, and as you may know by now, what genetics wants and needs is variability. So those two dogs with flat noses are probably going to have similar health issues, which will also be passed down to their babies. But selective breeding hasn't just been used on animals. It has also been used on plants. The principle is the same. Plants suffer mutations as well, which means that every now and again they acquire new traits that might or might not be beneficial for us. And when we find plants with traits which are beneficial for us, then we make sure we perpetuate that trait. The main traits that we pursue in plants are, for example, crops which have better yields, crops which present some resistance to certain pests, different colors and shapes for ornamental plants, etc. But as with animals and their congenital diseases, when plants in a crop all become genetically similar, other issues might arise. One very famous example of this was the Great Famine in the 19th century. This occurred in Ireland, and it was due to the fact that farmers were only using one or two high-yielding strains of potato. The problem was that the crops were all infested by a fungus-like pathogen, which ended up killing all of the crops, as they were all genetically similar. This left a lot of people without food on the table. It is estimated that this famine killed around 1 million people. But it's not all bad, and it is through selective breeding of plants that we now have certain foods such as broccoli, cauliflower, kale, etc. Well, maybe not so good after all. But selective breeding is a lengthy process. It can take several plant generations to reach the perfect result, so we came up with faster ways to do the same job. Instead of waiting for nature to do the work, we decided to intervene and create the mutations ourselves. With genetic engineering, we are able to choose which mutations we want certain plants to have, and we are able to do this much faster. These are called GMOs, or genetically modified organisms. Of course, this is a simplified explanation of what happens, but the reality is that using GMOs is easier, faster, and more likely to yield the results that we want, without having to wait for mutations to occur naturally. The most researched traits for GMOs are resistance to certain pesticides, so that way plants can be sprayed with pesticides and nothing will happen to the crop, only the actual pests. Other sought-after traits are crops with bigger yields and also crops which are more resilient to weather changes. So now let's talk about the genome. The genome is the whole genetic material of a given organism. So your genome will be different from my genome. Unless you're my identical twin that I don't know about, in which case your genome is exactly the same as mine. Because genetics is such an awesome and advanced discipline, scientists have actually been able to sequence the human genome. This means that they have been able to determine the sequence of genes in the human genome, so the sequence of the nucleotide bases. However, they found out that not all of our genome codes for proteins. Parts of our DNA are actually non-coding, so they seem to have no purpose at all. In fact, it is estimated that only 1% of the human genome actually codes for proteins, with the remaining 99% being non-coding DNA. However, with time, we have realized that there is a purpose to these non-coding areas of the DNA. It is now proven that they are actually involved in protecting against chromosomal deterioration during replication, they aid the protein synthesis process, and they may be involved in the formation of certain RNA molecules. There is still a lot we don't know about genetics, still a lot to be done and studied. But it is indeed one of the most promising and important areas of biology, and one that is bound to be developed even further in the future. I hope you have enjoyed today's video. Stay tuned for more exclusive content, and I will see you again next week. Bye bye!